Welcome to Weekly Neurology Deep Dive. Just a quick reminder, this deep dive is for educational purposes only, and it's really just a summary of the research. You'll want to read the full paper to really get into all the details in this episode.
Yeah.
We are going to be diving into a paper called Multiple Sclerosis, a Virus-Induced Gliopathy.
Mm-hmm.
It was published in Neurology, Neuroimmunology, and Neuroinflammation in early 2025 by Sabatino and Steinman.
Okay.
And, as neurologists, we know that, pinpointing the exact cause of MS has been a challenge. But, for a long time, the focus has been on the role of autoreactive B and T cells attacking myelin.
Right.
But this research really points to a potential interplay between a very common virus and specific antibodies and damage to astrocytes.
Yeah.
So are you ready to kind of explore this intriguing hypothesis?
Absolutely. Let's dive in.
All right, so the paper starts by acknowledging the kind of traditional autoimmune perspective of MS, but they introduce a protein called glial-cam.
Right.
What is the significance of glial-cam in this context?
So glial-cam is found at astrocyte junctions, and the key finding here is that researchers have discovered higher levels of antibodies against glial-cam in patients with MS, and this suggests that glial-cam might be an autoantigen, meaning the immune system is mistakenly attacking it.
So glial-cam emerges as a potential new target.
Exactly.
Now let's bring in the Epstein-Barr virus, or EDV, which we know has a very strong link to MS risk. Can you remind us about that connection?
Sure. So the link between EBV and MS is well established. Studies show that nearly 100% of individuals with MS have been infected with EBV at some point. Additionally, antibodies to an EBV protein called EBNA1 are elevated in MS patients, and this is particularly true in those with the HLA-DRB1 star 1501 risk allele, which, as you know, is strongly associated with MS susceptibility.
Okay, so we have this potential new target, glial-cam, and we have this very well-established link between EBV and MS. How does the research connect these pieces?
This is where the concept of molecular mimicry comes into play. Antibodies produced against a viral protein. In this case, EBNA1 can sometimes mistakenly attack human proteins that resemble them. And interestingly, glial-cam shares a region of structural similarity or homology with EBNA1.
So the theory is that the immune system, after fighting off EBV, might misidentify glial-cam as a threat because of this similarity and then launch an attack on astrocytes.
Exactly. It's like the immune system gets confused and attacks a friend instead of a foe.
Wow.
What's even more fascinating is that the researchers didn't stop at glial-cam. They also found increased antibodies to another astrocyte protein called MLC1 in MS patients.
Okay, so that definitely adds another layer. What's the connection between MLC1 and glial-cam in this scenario?
So MLC1 is a binding partner of glial-cam, so we might be witnessing a sort of domino effect here. The initial attack on glial-cam triggered by those cross-reactive antibodies could then lead to a subsequent attack on MLC1. This is a phenomenon known as intermolecular antigenic spreading. So think of it as a chain reaction. The immune system already attacking glial-cam starts targeting its partner MLC1, widening the scope of potential damage.
So it's not just a single protein under attack, but potentially a cascade of immune responses.
That's the hypothesis. This research really emphasizes the potential role of EBV infection in triggering an immune cascade that leads to MS. It presents very compelling evidence connecting EBV to the development of antibodies against not only glial-cam, but also MLC1 and even myelin proteins.
The evidence for EBV as a necessary but not sufficient factor in MS development is indeed very robust. The paper cites a 2022 study by Bjornevik and colleagues published in Science, which investigated a cohort of over 10 million young adults in the U.S. military.
And that study found that individuals infected with EBV had a 32-fold increased risk of developing MS compared to those who were not infected.
A 32-fold increase. That's huge. That really underscores the need to understand how EBV might be contributing to MS.
Yeah, and the paper highlights how the genetic risk factor for MS, the HLA-DRB1 star 1501 allele, further amplifies the risk associated with EBV infection.
Right.
A 2009 study by Sundstrom and colleagues in the Journal of Neuroimmunology demonstrated that individuals with this allele who are infected with EBV have particularly high levels of antibodies against specific domains of EBNA1.
So the presence of both EBV infection and this particular genetic allele creates a predisposition for MS development.
And that combination might be what drives the production of these cross-reactive antibodies that target not only EBNA1, but also glial-cam MLC1, and potentially other proteins involved in MS pathology.
The paper also highlighted a couple of other proteins targeted by these cross-reactive antibodies in MS patients. One was anoctamin 2. What is the significance of this protein?
So anoctamin 2, also known as ANO2, is involved in regulating ion transport and cellular excitability. And a 2019 study by Tengval and colleagues in PNAS found that antibodies against ANO2 were elevated in MS patients and that these antibodies cross-reacted with EBNA1.
So a gliocam ANO2 shares a region of homology with EBNA1.
Yes, this further supports the concept of molecular mimicry. The immune system primed by EBV infection produces antibodies that inadvertently target these critical proteins in the nervous system.
The other protein mentioned was alpha-B crystalline, or CRIAB. What role might it play?
So CRIAB is expressed in various glial cells and has neuroprotective properties. A 2023 study by Thomas and colleagues in Science Advances showed that antibodies against CRIAB cross-react with EBNA1 and are elevated in MS patients. What's interesting is that CRIAB also exhibits characteristics of an immune checkpoint inhibitor, a molecule that helps regulate the immune response.
So attacking CRIAB could potentially lead to more widespread inflammation in the central nervous system.
It's like removing a safeguard, which can allow the immune system to become overactive.
The researchers seem particularly interested in the connection of both gliocam and anoctamin 2 to water and ion channels.
Right, and disruption of these channels and astrocytes could significantly impact neuronal function as, astrocytes play a crucial role in maintaining the balance of ions and water in the brain, which is essential for neuronal signaling and overall brain health.
So this means that damage to astrocytes and disruption of these channels could contribute to the neuronal dysfunction and the clinical symptoms we see in MS.
It's a plausible hypothesis that requires further investigation. The authors propose that the disruption of water and ion channels might provide a new perspective on the immune attack in MS.
The paper also mentioned that gliocam shares molecular similarities with proteins found in the myelin sheath, contactin, and neurofascin.
Right, and this suggests that the immune attack in MS might not be confined to myelin, but could involve a broader attack on various components of the nervous system, including astrocytes and possibly even neurons.
This concept of a gliopathy fueled by EBV infection is a significant departure from the traditional understanding of MS as primarily an autoimmune attack on myelin.
It challenges us to reconsider our assumptions and explore new avenues for understanding, diagnosing, and treating MS. It highlights the importance of interdisciplinary research, bringing together virology, immunology, and neurology to unravel the complexities of this disease.
What practical implications could this research have for neurologists?
Well, the most immediate implication is the potential for new biomarkers. If specific antibodies against glialcam MLC1 or these other proteins are consistently elevated in MS patients, we could use them to diagnose MS earlier and more accurately.
That would be a huge advancement, especially considering the challenges currently associated with diagnosing MS. It can be a long and difficult process of elimination for patients.
Absolutely. And these biomarkers could also help us monitor disease activity and treatment response. We could track antibody levels over time to assess the effectiveness of therapies in controlling the immune attack.
That would allow us to really personalize medicine and tailor treatment strategies based on individual antibody profiles and potentially intervene earlier to prevent further damage.
Precisely.
The paper briefly mentioned potential treatment approaches such as monoclonal antibodies or other immunotherapies that could specifically target these pathogenic antibodies.
Another possibility is to develop therapies that enhance the function of these targeted proteins or protect them from immune attack, for example, boosting the expression of glial-cam or MLC1, or developing drugs that shield them from antibody binding might mitigate the damage.
The potential for cell-based therapies was also mentioned.
Imagine developing therapies that replace damaged astrocytes or enhance their ability to repair and regenerate.
These are exciting possibilities. But of course, much more research is needed before these therapies can become a reality.
Of course, we need to fully understand the role of these antibodies in MS pathology, determine their specificity to MS, and carefully evaluate the safety and efficacy of any potential treatments.
This has been a fascinating discussion. It seems like this research has opened a Pandora's box of questions and potential avenues for future investigation. We encourage you to explore the full paper for a deeper understanding of the findings. This research offers a lot to consider. It suggests that the immune attack in MS might be much more complex than we previously thought.
Yeah, it really underscores the interconnectedness of, different systems in the body. A viral infection like EBV, could trigger a cascade of events that ultimately leads to damage in the central nervous system.
The paper emphasizes the need for further research, but it also hints at potential therapeutic strategies that could emerge from these findings.
Absolutely. If we can identify the specific antibodies driving the attack, on astrocytes and myelin, we might be able to develop targeted therapies to neutralize them.
The possibility of using monoclonal antibodies or other immunotherapies to target these pathogenic antibodies was mentioned.
Right. And another intriguing avenue is the development of therapies that enhance the function of the targeted proteins or protect them from immune attack. Perhaps we could boost the expression of gliocam or MLC1 or develop drugs that prevent antibody binding.
The potential for cell-based therapies to replace damaged astrocytes or enhance their repair and regeneration capabilities is also pretty fascinating.
Yeah, these are all promising areas for future research, however. We must proceed with caution and conduct rigorous studies to ensure the safety and efficacy of any new treatments.
This research really encourages us to view MS through a new lens, considering the role of astrocytes, viral triggers, and the complexities of the immune response.
It reminds us that our understanding of MS, as with many diseases, is constantly evolving. Staying informed about these emerging concepts and critically evaluating new research is so crucial for providing the best possible care for our patients.
Well, this deep dive has been a really insightful exploration of a novel perspective on MS, and it highlights the interconnectedness of various biological systems and the potential for groundbreaking discoveries that could really transform the way we diagnose and treat this complex disease. We encourage all of you to delve into the full research paper to gain a comprehensive understanding of the findings and their implications for the future of MS research and clinical practice.